Today's Fun Fact Friday is Barbara McClintock from 1902 to 1992. Geneticist Barbara McClintock completed revolutionary work in the 1940s and 1950s that led to her discovery of genetic transposition, or jumping genes. However, her discoveries initially met with skepticism from her colleagues, and it took decades for technical advances in molecular biology to prove that her work was valid. In 1983, she was recognized for her astounding work, receiving the Nobel Prize in Physiology and Medicine for her achievements. McClintock's groundbreaking work began while she was a graduate student at Cornell University, where she was part of a group studying maize genetics. At that time, little was known about DNA, and most genetic studies focused on chromosomes and genes. The scientists at Cornell knew that several of the genes that determined kernel color. They also determined how kernel colors are predictably inherited. Thomas H. Morgan had experimented with fruit flies and found that chromosomes carried the genetic material and that genes are arranged in a linear fashion, along the chromosomes like beads on a string. He received the Nobel Prize for this work in 1933. Scientists had also learned that the chromosomes came in pairs, one from each parent, usually attached near the center to make an X. The paired chromosomes could recombine, trading genes from one pair to the other, but it was believed that the gene generally stayed on the same chromosome and in the same linear order. McClintock made her most valuable discovery while doing research at Cold Spring Harbor on Long Island. She carried out her experiments by growing corn in a garden plot behind her lab. Fertilization was controlled by covering the flowers to protect them from random pollinization, and they were dusted with pollen from a known source. McClintock found the maize exhibited an unexpected pattern of colored and multicolored kernels that couldn't be explained by traditional genetic theories. She believed that the genes controlling the kernel pigment had transposed or jumped to another location in some cells. Her results were largely ignored for years, as they contradicted genetic theories of the time. 50 years later, we now understand that there are indeed transpositions, genetic elements that can pop out of the chromosome and reinsert somewhere else, which activate or prevent the expression of certain genes. These transposable elements have been found not only in corn and other plants, but also in fruit flies, bacteria, and yeast. Barbara McClintock, 1902 to 1992.